Hello students, welcome to Nucleus Neet Foundation. How are you? कैसे हो आप? Are you wondering why I am wearing mask today? Because as you know, today's topic is virus. So few years ago, you all know about the incident, students, where COVID has shook the entire world. Many people died because of this COVID virus infection. So today's class we will discuss each and every point about virus and watch this video completely without skipping. So I am going to remove the mask. No need of mask now. So now let us enter into the topic. Virus. What is the meaning of virus? Virus means poisonous fluid or venom. Poisonous fluid or venom. Venom क्या होता है बच्चों? जहर, जहर. ये जहर का समान है. So it is equal to poison. The term virus means poisonous fluid, otherwise called as venom. Okay. So how actually is this virus particle? What is the structure of this virus particle? A virus particle, it is acellular. Acellular means it doesn't have any cell structure. It doesn't have any cell wall. It doesn't have any plasma membrane. There is no cytoplasm. There is no nucleus. There are no cell organelles. So you can say that there is no cellular structure. That's why viruses are considered as acellular organisms. They are acellular organisms. And they are obligate intracellular parasites. Obligate means compulsory, compulsory. Compulsorily they have to lead life of parasite. What type of parasite? Intracellular parasite. What is intracellular parasite? Intra means within, under. Within the host cell, they will lead the life of a parasite. Intra means what? Within. Within the cell, within the host cell, they act as parasites. Okay. So other than parasite, they cannot live outside the cell. They have to survive as a parasite inside the host cell. Okay. Once again, the definition, they are acellular, means they are not made up of cell structure, and they are obligate compulsorily. They are intracellular parasites. They live as parasites inside the cell. Okay. And this virus does not fit in Whitaker's classification. Why virus has no place in Whitaker's classification? Obviously, viruses does not have any cellular structure. They doesn't have any body which is made up of a cell. Okay. That's why they does not fit in Whitaker's classification. And the size, coming to the size, virus particles are extremely small. They are not even visible in the normal microscope. We have to use a special microscope called the electron microscope. The most sophisticated electron microscope only can show the structure of virus. You know that electron microscope will use electron beam, not light. It uses electron beam to magnify the submicroscopic particles. And what is the size? The size ranges from 10 nanometers to 300 nanometers. Extremely small, smaller than the bacteria, smallest of the smallest bacteria, smaller than even mycoplasma bacteria. Okay, extremely small particles they are. So they are measured in units called as nanometers, 10 to 300 nanometers. And they have inert crystalline structure. Their structure is crystalline. They have inert crystalline structure. You know what is a crystal? Salt crystal or sugar crystal? So they have structure of a crystal, just like a sugar crystal or a salt crystal, very minute crystal like structure. Okay. If you observe the body of virus, it has very simple body. That is, it has the outer protective covering which is called as capsid. What it is called? Capsid. And capsid is made up of which material? It is made up of protein. It is made up of protein. The outer protein covering is called as capsid. Inside the capsid, there is a very important molecule which is called the genetic material. The genetic material, it can be either RNA or DNA. The genetic material. The genetic material, it can be either RNA or DNA, but not both. कुछ वायरसेस में आरएनए होगा, कुछ वायरसेस में डीएनए होगा, दोनों एक वायरस में नहीं मिलेगा. समझ गया? Okay. So this is about the structure of a typical virus. Okay, so outside the living cell they are equal to non-living things. Inside the cells they behave as living organisms. That's why scientists consider these viruses as connecting link between living and non-living things. What is that? Connecting link between living and non-living things. Note down this point. Okay, viruses, they are very bad guys, students. They cause lot of diseases. They cause diseases in animals, plants, and also they will infect the bacteria. They can infect animals, plants, and also they are going to infect even bacteria also. Very bad, right? So what are the diseases caused by viruses? In humans, they cause diseases like flu, which is called common cold. सर्दी में होता है ना, common cold. Next, corona. You all know about corona, COVID-19. AIDS, very dangerous viral disease. इसको इलाज ही नहीं है. There is no cure for AIDS. Chickenpox, herpes, rabies. Rabies occurs because of dog bite. So जो कुत्ता होता है ना, पागल कुत्ता, the mad dog, when it bites, then this disease will be spread to the person. Actually the mad dog, a rabid dog, it behaves mad because of this virus, because it affects the nervous system, it affects the brain. Okay, so that's why be aware of this rabid mad dogs which are on the streets. So if you by accident, by chance, if you are bitten by these dogs, immediately what you should do? You should get vaccinated. Go to a government hospital which is nearby. They will give vaccine. Okay, if you ignore it, it will sometimes leads to death. So it is also a dangerous disease, rabies. Okay, so now let us discuss more points about virus. Let us discuss the discovery of virus. How scientists first discovered the virus. It was D.J. Ivanovsky, a Russian scientist. In year 1892, he recognized these virus particles as a poisonous liquid. So D.J. Ivanovsky, he was doing some experiment on a tobacco plant which was infected by a disease called mosaic disease. So this is a tobacco plant. This is a tobacco plant. It is infected by a disease. What is the disease? Mosaic disease. The mosaic disease is characterized by formation of several spots, white, yellowish spots on the surface of tobacco leaves. So at that time, Ivanovsky, he believed that this disease called mosaic disease was caused by some bacteria. Mosaic disease of tobacco plant was caused by some kind of bacteria. Thinking that it was caused by some kind of bacteria, he did one small experiment. What he did is, he separated the leaves from this infected tobacco plant and he extracted juice from the leaves. It is called as leaf extract. So he extracted the juice from the leaf. So that juice from the infected leaf is called as leaf extract. Then what he did, he tried to filter this leaf extract by using bacterial filters. Okay, so he took another beaker. In that beaker he kept a funnel. In that funnel he kept a filter. This is the filter. What filter is that? This is a bacterial filter. What is that? Bacterial filter. What is the function of a bacterial filter? It will trap the bacteria which is present in the leaf extract. So now what he did, he put this leaf extract in this bacterial filter and he allowed this juice to pass through this bacterial filter. So now the juice passed through this bacterial filter, drop by drop, he collected the filtrate. So once the leaf extract was completely filtered, he was so sure that some bacteria might have trapped in this bacterial filter. So what he did, he removed this bacterial filter He removed this bacterial filter and observed this bacterial filter under the microscope. When he observed under the microscope, surprisingly, he couldn't see any bacteria. There was no bacteria in the filter. No bacteria was found in that filter. So from this he came to conclusion that the disease mosaic of tobacco is not caused by bacteria but is caused by some agent, infectious agent, which is smaller than the size of bacteria. So what he found, he recognized some unknown microbe smaller than bacteria caused the mosaic disease in tobacco plant. Understood. Later, another scientist, M.W. Beijering, in year 1898, he demonstrated another interesting experiment. And what was that experiment? He demonstrated the extract of mosaic infected tobacco plant can cause disease in a healthy plant. So what he did, he took another tobacco plant which was healthy and fit. This is a healthy tobacco plant. He took small leaf extract. He took leaf extract from infected tobacco plant. Small amount of infectious fluid from this leaf was collected and that leaf extract was transferred onto a healthy plant. Then what happened? Immediately this healthy plant, it started showing the symptoms of tobacco mosaic. The disease symptoms developed in this healthy plant too. From this, Beijering, he came to know that the disease is actually caused by some kind of poisonous fluid. So he gave the phrase called as contagium vivum fluidum. He called that infectious material which is able to cause the disease in the healthy plant as contagium vivum fluidum. What is the meaning of this phrase? Infectious living liquid. Infectious living liquid. Contagium means infectious, vivum means living, fluidum means liquid. Okay, so this was another interesting experiment which was done by M.W. Beijering. Later, another scientist, W.M. Stanley, he was able to crystallize the virus particles. He crystallized the virus particles into small crystals. You can see here. These are the crystals of virus. Okay, these are not any other crystals of salts, sugar like that. These are the crystals of virus. The virus can be crystallized into this type of particles. Okay, so that was another achievement which was done by W.M. Stanley. He crystallized the virus and he also told that the crystallized virus largely it was made up of protein. Crystallized virus largely made up of which material? Protein. Note down this point in your notebook, students. Now let us discuss some more information. Next, viruses were first discovered by scientist D.J. Ivanovsky. The term virus was coined by scientist Pasteur. What is the meaning of virus? Virus means poisonous fluid or venom. So Pasteur, he gave the name virus to this infectious particles. What is the meaning of virus? Poisonous fluid or venom. Okay. So now let us see the classification of virus. A scientist called Holmes, he classified viruses into three types based upon the host organism which they infect. The viruses which are attacking the plants, are called as phytophages. The viruses which attack the animals, are called zoophages. The viruses which attack bacteria are called phages. They are just called as phages. Okay. Now what are the examples of phytophages, plant viruses? TMV, tobacco mosaic virus. Write down. PMV means what? Potato mosaic virus. CAMV, cauliflower mosaic virus. So tobacco mosaic virus, it infects the tobacco plant generally. Potato mosaic virus, it infects the potato plant. Cauliflower mosaic virus, obviously it infects the cauliflower. Okay, so next, animal viruses, zoophages, example HIV virus, polio virus, rhinovirus which causes the common cold, varicella which causes herpes, rhabdovirus which causes rabies. These are some examples of animal viruses. Next, the viruses which attack bacteria phases, T2 bacteriophage, 5174 bacteriophage, QB bacteriophage. So these are the viruses which attack the bacteria. Next, another classification of virus. Virus can be also classified based upon the genetic material, whether DNA or RNA. If RNA is present in the virus as genetic material, such viruses are called as RNA viruses. So in RNA virus, what genetic material will be there? There will be obviously RNA. RNA genetic material will be present in the virus. RNA viruses, again two types. Single-stranded RNA virus, double-stranded RNA viruses. So only one strand of RNA is present like this. Then that virus is called single-strand RNA virus. When two strands of RNA are present, inside the virus, it is called a double-stranded RNA virus. Okay. Next, coming to the DNA viruses. A DNA virus will have deoxyribonucleic acid. Deoxyribonucleic acid, DNA. What is full form of RNA? Ribonucleic acid. So the DNA viruses, again two types, single-stranded DNA viruses, double-stranded DNA viruses. A single-stranded DNA virus will have a single strand of DNA, whereas a double-stranded DNA virus have two strands of DNA. Okay, so students, now we will see different shapes of virus. Virus occur in different varieties of shapes like spherical shape, rectangular shape, bullet shape, polyhedral shape, rod shape, tadpole shape. So these are the different shapes of viruses. I repeat, spherical shape, rectangular shape, bullet shape, polyhedral shape, rod shaped, tadpole shaped. These are some general shapes of viruses, and these viruses, they have genetic material either RNA, ribonucleic acid, or DNA, deoxyribonucleic acid. Okay, so now let us see how a virus can infect its host cell. You know that viruses are host specific. They will attack only a particular host. A tobacco mosaic virus will attack the tobacco plant. A cauliflower mosaic virus will attack only the cauliflower. So this is called as host specificity. The plant viruses will not attack the animals, and animal viruses will not attack the plants. Understood? Now let us see how a virus generally attacks its host cell. Okay, this is a virus particle which is just outside the cell. When a virus is present outside the cell, it is an inert particle. It is an inert crystalline particle. It does not show any signs of life. Okay, it is equal to a non-living thing, a non-living particle. So when the moment the virus come into contact with the surface of cell, the virus particle, it attaches to the plasma membrane of the host cell. Then slowly this virus particle, it gains entry into the cytoplasm. It will enter into the cytoplasm of the cell. When the virus enters into the cytoplasm, it will release its genetic material. It will release its genetic material into the cell. When the genetic material is out, then what happens? The genetic material takes entire machinery of the cell into its control. Previously, the cell is under the control of nucleus. Now, the genetic material will take the entire control of the cell into its hands, and it will order the cell to synthesize new virus particles. Now, the cell can't do anything. The cell simply shuts down all other important activities and it will obey the viral genetic material. So, according to instructions of viral genetic material, our cell now, poor cell, what it does? It has no another option, so it has to obey the genetic material and it will produce new virus particles. So sad. So new virus particles are produced in large numbers, hundreds of virus particles, thousands of virus particles will be formed inside the cell. When the cell gets completely stuffed, packed with the virus particles, it bursts. When it bursts, then these virus particles, all of a sudden they come out. All the virus particles, they come out of the cell like this. And again these viruses are ready to infect other healthy cells which are around. Like this virus will spread from one cell to another cell and they keep on damaging all the cells. And because of that, we will feel the symptoms. We'll feel the symptoms of virus infection like fever, okay, weakness, nausea, headache. This type of symptoms we feel, students. Okay, so antibiotics does not work to treat viral infections. Antiviral drugs can be used to treat these viral diseases. You know, the common cold generally which we get during rainy season, it is one of the viral disease which is caused by a virus called rhinovirus. Rhinovirus causes common cold. It infects the upper respiratory tract. We will get sneezing, we will get running nose because of this virus. Okay. So however, our body is having a good defense mechanism. Our body has immunity. So our immune system will fight against this virus and they will damage the virus particles. Soon again we can restore our health. But in case of some viruses like HIV virus, our immune system cannot tackle the HIV virus. So our body surrenders to this virus and the virus dominates and it causes death in certain cases. Okay. So the virus infections can heal by themselves without using any medication because of our immune system. But virus like HIV virus, there is no cure. Our body cannot deal with that virus and there is no medicine also. Okay. So now we will discuss what are the symptoms. What are the symptoms of viral infection in the plants? When plants get infected by virus, then plants do also show some symptoms. What are the symptoms? The plant viruses cause symptoms like mosaic formation. Mosaic formation, you can see the picture. The leaf develops a lot of spots on the surface which in the pattern of mosaic, it is called as mosaic formation, as you can see in the tobacco mosaic disease. Okay. Next is leaf rolling. The leaf rolls on itself. Okay, this is another symptom, leaf rolling. Next, leaf curling. The leaf becomes deformed. The leaf becomes deformed and shapeless because of viral infection, as you can see in this picture. This is called leaf curling. Next is leaf yellowing. The leaf turns into yellow color because of loss of chlorophyll pigment. This is another symptom. Next, vein clearing. The veins which are present in the leaves, they just become colorless. They become white because of loss of chlorophyll. This is another symptom of viral infection in the plants. Dwarfing. The plant becomes short. It cannot grow to its normal size. That is called as dwarfing, short height. Stunted growth, reduced growth in the plant. These are all the symptoms of plant viral disease. Very important, students. Next, these 4 points are very important. Listen carefully. Most plant viruses, जो भी plant viruses है, जो plants को infect करेगा, वो viruses सारे के सारा single stranded RNA is present in general. In those viruses, you can see single stranded RNA as genetic material in general. Okay. Next, animal viruses जो भी है, इस animal viruses में ssRNA या double stranded RNA या double stranded DNA पाया जाएगा. We can find either ssRNA, dsRNA or dsDNA in animal viruses. General bacteriophages, those viruses which attack bacteria, that is bacteriophages, they have double stranded DNA in general as genetic material. These 3 points very important for your NEET exam. Next, RNA viruses are fastest in evolving process. So RNA viruses, they evolve very fast when compared to any other viruses. Why? Because RNA is a unstable genetic material, it changes very fast. As a result, these viruses also can evolve into new strains, and that was one of the reasons why the COVID virus, it was able to produce several strains in very short period of time. Okay, so note down these points, students. Note down all these points. So once you have noted down these points, now listen this. What are the examples of single-stranded RNA virus? What is a single-stranded RNA virus? Inside the virus, there will be a single strand of RNA as genetic material. Example is your tobacco mosaic virus, retrovirus, HIV virus. Double-stranded RNA virus. This virus have two strands of DNA. Two strands of RNA. Example, reovirus, wound tumor virus. Next, single-stranded DNA virus, there is only one strand of DNA in this virus. Example, 5-174 bacteriophage virus, M13 phage. These are bacteriophages. Next, double-stranded DNA virus. Examples, cauliflower mosaic virus, lambda phage virus, adenovirus. These viruses have two strands of DNA, double-stranded DNA. Okay. So this is about viruses, students, copy down. Students, now TMV virus, tobacco mosaic virus, we are going to discuss the diagram, the figure of this virus. When you observe this tobacco mosaic virus under electron microscope, these virus particles, they look like long needles like this. They look like long needles under the electron microscope. Okay, each virion of tobacco mosaic virus, it looks like this. It is having the body which is cylindrical in shape. At the center of this cylindrical body, there is a hollow space which is called as core. In this hollow space, there is spirally arranged single-stranded RNA as genetic material. So attached to the single-stranded RNA, there are the protein subunits called as capsomeres, which are attached to the spirally coiled single-stranded RNA. So overall, you can see the shape like this. The shape of the virus will be like this. The protein subunits, which are looking like grapes, okay, they will be all attached to the single-stranded RNA from all directions. So that gives an appearance of a cylindrical shape. Okay, so several of these individual protein units called as capsomeres, collectively they form a protective layer on the surface of the virus, which is called as the capsid. Understood, everyone? So what are the symptoms of this tobacco mosaic disease? It causes tobacco mosaic on the leaves. So the tobacco plant, it develops spots in the form of mosaic pattern. It is one of the symptoms of tobacco mosaic disease. Okay, so copy down this diagram, students. Now the another virus, bacteriophage. Bacteriophage, it infects the bacteria, prokaryotic cells. You can see here, this is a bacteriophage virus. What is the shape? It is having the tadpole shape. It looks like a tadpole. Okay, so this virus particle, this virion, it will attach firmly to the surface of bacteria. So bichara bacteria kuch nahi keh paega. This virus, now what it will do? It will make hole on this poor bacteria. It will make a hole on the poor bacteria by using its spikes, sharp spikes. After making hole, it will inject this genetic material. It will inject its genetic material into the bacterial cell. Once the genetic material is injected into the cell, then the cell wall will close. Then what happens? This genetic material of virus, now it will take the control of the entire cell, and it will order this bacterial cell to produce new virus particles. ये देखो नया वायरस पैदा हो जाएगा अंदर. new virus particles are formed in hundreds of numbers. Okay, once many daughter bacteriophages are formed, the cell will burst, the prokaryotic cell will burst. ये जो वायरस अंदर है वो सब बाहर आ जाएगा और ये इन्फेक्ट करने लगेगा दूसरा बैक्टीरिया सेल को. So इस तरह, in this way, these bacteriophages will kill the bacteria. Okay, so this is about bacteriophages. Bacteriophages are not at all harmful to plants and animals. They will only attack the bacteria. You can see the diagram of bacteriophage from NCERT textbook. This bacteriophage is having head which is made up of the protein. Inside the head there is genetic material, DNA, and below the head there is a disc shaped structure which is called as the collar. Below the collar there is a cylindrical structure which is called contractile sheath. At the bottom you can see leg-like structures which are called as tail fibers, by using which the bacteria can attach firmly to the cell wall of bacteria. So copy down this diagram, students. Diagram of bacteriophage. Students, now we will discuss another type of infectious agents which are called as viroids. What are viroids? Viroids are the naked infectious RNA molecules of low molecular weight, and they were first discovered by scientist T.O. Diner. Look at this diagram. This is a RNA molecule. RNA means ribonucleic acid. The RNA molecule having low molecular weight, and it is infectious. That means it is able to cause disease in other organisms, and it is not covered by any type of protein covering, just as we see in the case of virus. And that is the reason they are called as viroids. Who discovered them for first time? Discovered by T.O. Diner. Remember, they does not have any protein coat, not covered by protein coat. And how these viroids can increase their number, how they can multiply? They replicate inside the plant cells. They will use the machinery of plant cells in order to produce new viroid particles. Okay, and they move to adjacent cells via plasmodesmata. So in the plants, you know that the cells are mutually in contact with each other by connections which are called as plasmodesmata. So through these connections called plasmodesmata, the viroids can actually move from one cell to another cell and next cell to the adjacent cell. So in this way the viroids can spread throughout the plant body. Okay, so scientists, they believe that these viroids get transmitted from one plant to another plant through insects and also by use of contaminated agricultural implements. Mainly these viroids, they cause plant diseases. What are the different types of plant diseases caused by viroids? Spindle tuber disease. So this is a disease which is seen in potato, potato plant. You can see the picture of the potato plant, the potato which is infected by this particular viroid. Spindle tuber viroid causes spindle tuber disease in potato. The potato becomes long and spindle shaped because of this viroid infection. You can see the picture here. So next, another disease, citrus exocartis, which this is the disease which occurs in the citrus, that is the lemon trees, and cucumber pale fruit. The cucumber fruit will become pale because of viroid infection. So this is all about viroids. Now next, let us move on to another infectious particle, prions. Prions are the infectious protein particles. Just like viroids, these are also infectious. And how is their structure? Their structure is something like this. They are made up of polypeptide chain. They are made up of polypeptide chain like this. So the polypeptide chain, it undergoes folding in the form of beta pleated sheets. This is described something as the most stable structure of the protein that is resistant to the proteolytic enzymes and also high temperatures. So these prions, which are called as infectious protein particles, they are formed due to misfolding of proteins. They are formed due to misfolding of proteins, and they were first observed by scientist Prusiner. They are formed due to misfolding of proteins, that is improper folding of proteins. You know students, inside the cell, the place where the protein synthesis takes place, that is in the rough endoplasmic reticulum, in the ribosomes. After protein synthesis process has completed, next these proteins, they undergo maturation process in the Golgi complex. Their proper folding of proteins will take place. So sometimes what happens, the polypeptide chain, which is the chain of amino acids, the polypeptide chain, it doesn't fold properly because of this mistake, some errors in the folding process. What happens, this type of abnormal proteins will be formed and they will be infectious. They will be infectious to the living organisms. Mainly they will infect the animals. Okay, so they are resistant to high temperatures. Even at high temperature also, these prions will not get degraded. The enzymes cannot degrade these prions. Okay, now what are the diseases caused by these prions, infectious agents called as prions? They cause diseases like the Kuru disease of Vam tribes in New Guinea. So in New Guinea, there is a place called as Papua. In that place, there are some certain tribes who are living. They are called Vam tribes. These Vam tribes, these are the people who live in the forests. These people, aboriginals who are living in the forest, they have a strange ritual. So that is, they celebrate the funeral. The ritual is totally strange, that the entire family members of the deceased person, they are going to eat the brain of dead person. Okay, they will open the skull and they will eat the brain, all the family members. So it is considered as a kind of honor which is given to the dead person. Okay, you can see here the Vam tribe person, a man who is holding the skull. He is a Vam tribe. So when these people eat the brains of the deceased person containing prions, the prions enter into their body, and since they are resistant to the digestive enzymes, they won't get digested because they have very stable structure. Beta pleated sheet structures will be there, and because of that, the prions being undigested, they enter into the bloodstream. From bloodstream, these prions, they will reach the brain. So in the brain, they start producing new prions. So thereby what happens, the entire brain get affected. These people, they cannot stand properly. They will lose muscular control and laughter, continuous laughter. These are the symptoms of this Kuru disease, okay, which is seen in the New Guinea tribe people called as Vam tribes. Okay, so how this is caused? This is caused mainly because of the ritual of eating the brains of dead person who was already suffering with this disease called as Kuru disease. Next, another disease is CJD disease called Creutzfeldt-Jakob disease. It is a neurological disease where the prions are formed inside the brain and the nervous system will be affected. Next, mad cow disease. Here also the cattle like cows, buffaloes will be affected by prions. The prions which are formed in the brain, they are going to damage the brain. They are going to cause encephalopathy. Okay, so that's why it is called bovine spongiform encephalopathy. The nervous tissue in the brain, it becomes spongy. So that's why it is called bovine spongiform encephalopathy. Next, another disease is the scrapie disease of sheep. The sheep suffer with this disease called scrapie disease, which is also a neurological disorder which is caused by prions. So now we will discuss about plant kingdom and animal kingdom in brief. Okay, students, now we will discuss kingdom plant A in brief. Kingdom plant A includes eukaryotic chlorophyllous organisms. They are eukaryotes, a well-defined nucleus is present and they have chlorophyll pigment using which they can carry out photosynthesis to prepare their own food material. And few of these plants are partially heterotrophic. That means some of the plants are found to be heterotrophic. They depend upon other sources of food material. So what are those plants? Some are insectivorous, Some are parasitic plants. What are insectivorous? Insectivorous plants are those plants which will depend upon the insects for nitrogen nutrients. They will trap the insects by different mechanisms, and after trapping the insect, they are going to digest the insect thoroughly and they absorb the juices from the insect. So in the juices, they will collect the nitrogen rich organic materials. Okay, in this way, this insectivorous plants, they obtain the nitrogen nutrients from by killing the insects. They are called as insectivorous plants. Some are parasitic plants. Parasitic plants are also partially heterotrophic, such as the cuscuta plant. Cuscuta plant is a complete stem parasite. The plant does not produce any leaves. It simply grows on host plant and absorbs the nutrients from the body of the host plant. Okay. Next, another important character of plant kingdom is that presence of cellulosic cell wall. The cells have cellulosic cell wall, and cellulose is a kind of polysaccharide. Okay, it is a tough material which gives protection to the cell from external environmental conditions. And the plant cells show chloroplasts which carry out photosynthesis. And the plant kingdom includes different types of members such as the algae which grow in water, bryophytes which grow both in water as well as on land, pteridophytes such as your ferns, gymnosperms such as your pinus tree, angiosperms such as your mango tree which produce the fruits. So now, in the life cycle of plant kingdom, you can see two distinct phases. Sporophyte phase, which is a diploid phase, gametophyte phase, which is a haploid phase. Both the phases, they alternate with each other. From the diploid sporophyte phase, haploid gametophyte will be formed. From haploid gametophyte, again diploid sporophyte phase will be formed. Okay. Sometimes these two phases may depend on one another. Sporophyte in some plants may depend upon gametophyte for nutrients. In some cases, gametophyte may depend on sporophyte for the nutrients. Okay. So in the life cycle of plants, sporophyte and gametophyte, they will alter with each other. This we call it as alternation of generation. Look here. Sporophyte phase which is a diploid phase, it will give rise to gametophyte phase. Gametophyte phase. The gametophyte phase, once again it will give rise to sporophyte phase. Okay, this repeats like a cycle. Okay, sporophyte and gametophyte phases, they will alter with each other. So that's why we call this as alternation of generation. The plant kingdom members show alternation of generation. So what is alternation of generation? Sporophyte produces gametophyte and gametophyte again produces sporophyte. This process repeats on and on, again and again. We call it as alternation of generation. It is one of the important characters of plant kingdom. Understood? Now let us discuss in brief about kingdom Animalia. Students, now we will discuss the last topic of biological classification, that is kingdom Animalia. Kingdom Animalia, it includes heterotrophic eukaryotic organisms which are multicellular. Heterotrophs, that means they depend upon other source of food material and they are eukaryotic and they are multicellular. Their body is made up of many cells. Okay. Next, the cell wall is absent. Very important. Cell wall is absent in the animals. There is no cell wall. So how they manage without cell wall? If cell wall is absent, how they can maintain their body structure? There is a skeletal system to maintain the body structure and shape. Okay. Next, the third point. The animals indirectly or directly depend on plants for food. What is the meaning of this point? Indirectly or directly depend on plants. Herbivorous animals, directly they eat the plants. Carnivorous animals, they eat the herbivorous animals. Herbivorous animals in turn eat the plants. Okay. In this way, animals somehow indirectly they depend on plants for their food. Okay. So this is another point. Next, digestion occurs in an internal cavity. Digestion occurs in an internal cavity such as the stomach and intestine. This is another important point. The nutrition is holozoic nutrition. Holozoic is a type of nutrition under heterotrophic nutrition where the organism consumes the food into its body. The food gets digested inside the body. So this process of heterotrophic nutrition, we call it as holozoic nutrition. Okay. Next, the reserve food is glycogen and fat. Animals, they store the reserve food material in their cells in the form of glycogen. Glycogen is a polysaccharide and fat is a lipid. Okay. So whenever they are in process of starvation, when they doesn't have adequate food, at that time they use these reserve food materials called glycogen and fat to meet the energy requirements of the body. Understood? Next point, they follow a definite growth pattern by growing into adults that have definite shape and size. So the growth pattern is definite. They have a young age, juvenile stage. So from the juvenile stage, the animals enter into the reproductive stage. From the reproductive stage, the animals enter into senescent stage, and afterwards, at the end of senescent stage, they are going to die. Okay. So this is called what we call a growth pattern. A particular growth pattern is seen in the animals. They doesn't randomly grow into any different shape, strange shape. Okay. They have a particular pattern in their growth. Okay. So that is about the animals. Whereas in plant kingdom, we don't see a proper pattern of growth. So for example, a tree is there. One tree will produce 3 branches. Another tree may produce 4 branches. The shape of one tree will be quite different from another tree when they become adults. But in animals, it is not like that. When animals become adults, all the animals look one and same, more or less. Okay, that is the meaning of this sentence. They follow definite growth pattern by growing into adults and that have definite shape and size. Okay. Next, higher animals show elaborate sensory and neuromotor mechanism. Higher animals in the sense the well-developed animals, okay, which are the mammals you can take, okay, reptiles you can take. So in these higher animals, you can see elaborate sensory and neuromotor mechanism. The sense organs are well developed and neuromotor mechanism is also well developed, because of which they can show complex movements in their body. Next, mostly they show locomotion. Most of the members in animal kingdom, they show locomotion. Locomotion is a movement by which the organism can move from place to another place. Okay. Next, sexual reproduction takes place by process called copulation. By process called as copulation between male and female organisms. So what is copulation, students? The male and female organisms during the mating season, they come closer. Once they come closer, then the male organism is going to deposit the male gametes in the female reproductive system. The process by which the male animal is going to introduce its male gametes into the female reproductive system of a female animal, we call that process as copulation. This is another important method of reproduction which is seen in kingdom Animalia. So these are some of the important points. So this marks the end of the chapter biological classification. Let us celebrate. We have completed biological classification, students. We will meet in the another class with another chapter.